So here we're going to talk about muscle cells and basically how muscle contractions happen. But before we talk about the actual function of muscles, which is to contract and release, we need to talk about the function and how exactly it does that. So we start off with the muscle, and then muscle is made up of separate muscle tissues. Muscle tissues are made up of muscle fibers, also known as contraction. From there, we actually don't say that it's a cell because it's multinucleated, so we call it this name. See, here we have nucleus, and the membrane around it is called the sarcolemma. So then further from there, we have myofibril, which are these individual units going that way, and then finally we have the myofilament, which consists of actin, which is a thin filament, and myosin, which is a thick filament. So if we look over here, we have a transverse section of a myofilament where we see the thick filament myosin in red, and we see the black lines here, which are thin filaments known as actin. So from Z line to Z line is known as the sarcomere, there are many sarcomeres in a myofilament. This small section, which if you see only contains actin, is known as the I band. The A band, which is a darker portion of the sarcomere, contains both the thick filament myosin and the thin filament actin. And then finally we have another I band. The H zone, which is the lighter part of the sarcomere, only has the thick filament myosin. So if we look here, we have a thick filament myosin and we have our thin filaments, the actin. And as we see, there are many troponin molecules and tropomyosin molecules that are on the actin fiber. It's a fibrous protein. And here the myosin has heads. So before we get into how it contracts, we need to understand how it actually is instructed to do so. So as we learned before, we have neurons, and they will transmit action potentials from one to another through synapses. But here we actually have a specialized type of synapse. We have an intermuscular junction, which is when you have the presynaptic neuron connecting or transferring to a muscle cell, which as we learned is differentiated here. And this is known as the sarcolemma. So then the action potential will come from the neuron, and it'll come here, depolarize the membrane on the presynaptic neuron, and it'll cause calcium voltage-gated channels to open. This will cause calcium ions to diffuse down their electrochemical gradient, and it'll stimulate vesicles that contain the transmitter substance. In this case, it's acetylcholine. It'll cause the vesicle to move towards the membrane, fuse with it, and then finally release its contents through exocytosis. The acetylcholine, or whatever transmitter substance is being used, will diffuse across the intermuscular junction and will bind with the receptors that are found on the muscle cell, or the sarcolemma. This will cause sodium ion channels, not voltage-gated, just normal ones. It will cause them to open, which will cause the sodium ions to diffuse down their concentration gradient, and it will cause the depolarization of this membrane. So once it depolarizes, it causes an action potential within the muscle cell. So after this happens, the action potential will be relayed down. It can't be seen here, but it'll be relayed down the syncytium, and then it will pause it.